Well, hello everyone and welcome. Jesus Futures 2, The Future of Regenerative Medicine, is the second in a new series entitled #JesusFutures, where we examine future possibilities and explore important questions pertaining to a range of scientific topics and cutting-edge research. Tonight's event examines the future of regenerative medicine and the new Institute of Developmental and Regenerative Medicine at Oxford. It is my pleasure to introduce our speakers tonight. Professor Paul Riley is Professorial Fellow in Medicine at Jesus College, British Heart Foundation Professor of Regenerative Medicine, Chair of Development and Cell Biology in the Department of Physiology, Anatomy and Genetics at the University of Oxford, and Director of the Oxbridge BHF Center of Regenerative Medicine. Professor George Hollander is the Hoffman and Action Medical Research Professor of Developmental Medicine and Head of the Department of Pediatrics and Professorial Fellow in Developmental Medicine at Jesus College. In addition to his academic roles in Oxford, George is also the Director of the Botner Research Center for Child Health, where scientists jointly address essential challenges in global pediatric and adolescent health. Professor Shankar Srinivas is Zeitlin Fellow and Tutor in Medicine at Jesus College, Wellcome Trust Senior Investigator, and Professor of Developmental Biology in the Department of Physiology, Anatomy and Genetics at the University of Oxford. His research group is also part of a collaboration that has recently been awarded a major Enriching Engagement Project grant by the Wellcome Trust for Shaping Destiny: Experiments in Embodiment, a major cross-divisional collaboration between the Department of Physiology, Anatomy and Genetics and the Oxford Research Center in Humanities, or TORCH. So welcome to you all, and thank you so much for joining us. No strangers to Jesus. And shall I just start by turning over to you, Paul? Thank you so much.
Thank you, Brittany, and good evening, everybody, and it's a pleasure to be here. I thought we would just basically start off by perhaps elaborating a little bit more on the introduction around some of our specific interests and associations with the college, for example. So in my case, I work on cardiovascular research. I've been working with my own laboratory for the last 20 years, and I was previously in London. In 2011, a British Heart Foundation professorship award brought me to Oxford when I was made a professorial fellow at Jesus College, which was a great pleasure and a privilege. And in that time, we work on heart development. We model congenital heart disease, so birth defects that arise through the heart. And then we extrapolated some of that work into how we might repair a heart after injury. And we've worked on heart attack as a model system and how we might restore damaged tissue during that process. And so the work that we're currently doing is moving in a direction of working with animal models, but also with human tissue and human cells to try and translate some of our findings. And in particular, we've been very interested in drug discovery, in working out ways to restore embryonic or developmental potential to adult cell types that are found in human hearts that might help to restore lost tissue after a heart attack. So that's me, and if I hand over to George Hollander to elaborate a little bit further on George's interests.
Good evening, everyone. Apologies for that. I'm by training a pediatrician. I had my initial medical school training in Switzerland. I'm Swiss, and then had further training in experimental immunology, both in Switzerland and in Boston, and joined Jesus College 12 years ago when I relocated to the United Kingdom. The research that I had been doing for many years is focusing on how the immune system develops. And this obviously links to pediatrics, and the research that we are undertaking is focused on such questions as how are we as an individual able to recognize our own tissues as self and don't with a while as soon as we see cancerous cells, bacteria, viruses, parasites, we can mount a protective immune response. And this is an active learning of the immune system that changes also with age. And so in this context, immunology is extremely dynamic and is undertaking learning as we go along. And this learning is relevant if you think about organ regeneration, as we will talk in a couple of minutes, or if you think about transplantation. And so my research group will continue at the new institute, going after exactly the questions, how the immune system learns and develops this capacity to distinguish between self and non-self. Shankar.
Thank you, George. I'm a basic scientist, a fundamental scientist by training. I did my undergraduate training in India in Nizam College, and then moved to New York, to Columbia University, where I did a PhD on how the kidney develops, studying the developmental biology of the kidney. Then moved to London, where I'm shifted slightly in terms of experiment questions to how the entire body is shaped, how the head-tail axis is set up. And then I moved to Oxford in 2004, set up my own research group here. And we continue to focus on how the body is shaped. That's the fundamental question that we work on. We arise, all of us, from a single cell, the fertilized egg, which of course has no shape. It isn't as though there's a preformed tiny homunculus within this fertilized egg, which just grows in size. It looks nothing like a body. And it's a very complicatedly and precisely choreographed cell movements that shape the body. And that's what my research group focuses on. And so, of course, to be able to understand this and access this, one of the main technologies we use is imaging, to be able to see how these shapes emerge during embryonic development. So my group, in terms more specifically, the areas that we work on are pretty early during embryonic development. So shortly after implantation, the stages that would be equivalent to in humans, the first 3 to 4 weeks of embryonic development. And we look at how differences are generated in terms of the front of the body, the back of the body, the left and the right, etc. And the related question of how the heart, which is is arguably the earliest organ to form, the first solid organ to form after the blood, and how the heart forms and is shaped. So, and then in addition to all of that, of course, I had the pleasure of being the medical fellow in Jesus College and tutor the medics. And I think I see one former medic here on the call, which is really very pleasing. It's good to see you, Natalie. So I'll hand back to Paul.
Thanks, both. So just to begin with, in terms of introducing the sort of concept of regenerative medicine, we like to quote an Oxford alumnus, Nobel Prize winner, back in 2002, Sidney Brenner. Apologies that he was actually an alumnus at Exeter College, but never mind, we'll let him off for that. But he came up with a very nice definition that regenerative medicine is the process of replacing or regenerating human cells, tissues, or organs to restore or establish normal function. And it comes in various flavors. There are different ways to approach the problem of trying to restore lost tissue after disease or injury. There are approaches that involve cell therapy, so injecting or transplanting cells into diseased or injured tissues with the hope that they might engraft, survive, and give rise to new tissue or signal to improve the environment or the actual injury response. There is bioengineering, tissue engineering, the making of smart materials. They could be biological products or actually synthetic products which help patch up an area of damage and invoke a nice response that will restore function to the organ. There is gene therapy, in which the introduction of genes to give rise to proteins that can activate resident cell types within organs to actually promote repair. And there is a drug-based small molecule type therapy in which you can stimulate the resident cells by drug addition to drive the process of repair and regeneration. Now, the new institute, just to flag immediately where we are with that, is based primarily on the idea that we would stimulate cells within tissues and organs, and we would drive developmental programs to restore what happens in the embryo during development to give rise to new tissue, etc. So that's the principle of the Institute, just to bring that in straight away. But to give you a couple of sort of examples where regenerative medicine has been very important, and if I'm allowed to, I'll just mention our own work in which we've made discoveries around how a cell type around the outside of the heart called the epicardium, in which humans have an epicardium, mice have an epicardium, even zebrafish have an epicardium. It's highly conserved. This cell type is very active in development. It gives rise to the parts of the coronary blood vessels and the scaffold of the heart, the fibroblasts, and even to some heart muscle cells. But it goes dormant. It goes basically quiescent, asleep during adulthood. And we found a way to reactivate the dormant cells in the adult heart to behave like they do in the embryo. And they gave rise to new blood vessels and indeed some muscle, which helped significantly in animal models of heart attack to improve outcome. So that was just one finding from our own work. And we're now trying to seek drugs, etc., that could stimulate that process further. And we're testing those in human epicardial cells from patient biopsies as a screening program that will move into the new institute. And then just to give you a second example that is nothing to do with our own work, there's a very interesting study in Japan in which pluripotent stem cells were used to derive retinal pigmented epithelial cells, RPE cells, placed into a sheet of tissue, and these were transplanted onto a woman patient who had macular degeneration or loss of visual acuity in the retina. And that tissue sheet restored a lot of the lost vision in that individual patient. There are lots of issues with that and using that approach, not least of which the cells, whether or not they elicit an immune response, and George knows much more about that than I do, and how you produce those in readiness for a patient at scale in order to manufacture a product. But at least in a N=1 patient cohort, this seemed to be quite effective. So a very nice example of going straight into a patient with a very severe retinal disease. So those are just a couple of examples. And maybe I could hand over to George just to elaborate a little bit further on regenerative medicine in general terms.
Yeah, thank you very much, Paul. So the example that I wanted quickly to talk about, because it touches upon the research in my lab, is the ability, or sometimes the need, to regenerate the immune system. And that obviously comes into play when you have somebody who requires a bone marrow transplantation because of a malignancy in the immune system or in the blood system itself. And this is probably one of the examples where medicine has been, in the regenerative space, extremely successful, although there are a number of hurdles still to be taken. So basically, where you can now eliminate the entire blood system of a patient that has, for example, leukemia, and then come in with the stem cells. You don't even have to put the stem cells back into the bone marrow where they typically reside, because they find their way themselves. So you just get an infusion of these precursor cells. They will home to the bone marrow, find their niches where they are happy and where they grow, and where they recapitulate the developmental program that they have learned and established during embryonic development. And while this works even in elderly individual like myself, I had 12 years ago a bone marrow transplant, it still requires that in a regenerative effort, this process is supported. It's supported by making sure that the cells are most effective in regaining the different cell types and their relative and respective functions. And so work that will go on in the space of bone marrow transplantation is work that is related to creating these niches where these precursor cells sit, to accommodate them better, and it creates efforts that allow these cells, while they develop, to develop faster, particularly in old age, and to develop as normal as they would and have during embryogenesis. The second example that as a pediatrician strikes me always just to be absolutely amazing, and here we just know it works, but we don't really know why it works yet, is the fact that in early development, if you break the skin, for example, you can regain the intactness of the skin without a scar. So you regenerate your normal anatomy and structure of the skin without a scar. Shortly after birth, you lose that. And as we all know, the older we get, the longer it takes to build the scar, and typically the scar as nicely structured as it was when one was much, much younger. And here again, the process of regeneration is in play. It draws on learned and established rules and regulations, but it is not as effective anymore as it used to be. And so the formation of a scar can occur not only in the skin, but in many other organs as well. And part of the Institute will also look at how this scar formation occurs and what we can do that instead of a functional tissue, that instead of a scar, we regain functional tissue. So these were, I think, two examples that relate to the Institute, that relate to the research that my group does, and in part to my own biography. But Shankar.
Thank you, George. I just wanted to follow on from George to actually emphasize how much of our advances in regenerative medicine are founded on pretty fundamental research. And so one of the classic examples, which we were actually just talking about earlier, is take the Drosophila, the fruit fly, as a model system. Sometimes you can do experiments. As scientists, we do experiments on a variety of model systems. And there are mutants in the fruit fly which don't have a heart. They have a severe heart defect. And in fact, the particular mutant in the fly that I'm thinking of is called a Tin Man, after the Wizard of Oz, where the Tin Man doesn't have a heart and is in search of a heart. And it's remarkable. The very same gene in the fruit fly that is damaged in the case of the mutant, which doesn't have a heart, there are mutants in that gene in humans that have been, mutations have been found in humans with cardiac defects of various sorts, indicating how there's a conservation of function, fundamental conservation across very, very large separations in relatedness between animals. So something as distant as a fruit fly and a human has this fundamental conserved function in this gene. It happens to be also called NKX25 in humans and mice. There's a remarkable conservation of these functions, and so you can use model systems to address these type of issues and learn from experiments on model systems. We in my group, we use the mouse predominantly, but we are also doing work on the human. And in the IDRM, we will continue to use mouse, the human, and there are a variety of other model systems. Paul and George will describe theirs. And in fact, Paul and I are jointly part of a large consortium funded by Wellcome to open up research in human embryos, open up the possibility of the community doing research in this field. And this is an area that the UK is a leader in. because of the availability of samples and because of several groups that have been able to use these samples to do cutting edge research to understanding humans. Because having said all of this time how models are really important and can be conserved, of course, some things it's always best to look directly in humans. And it's always good to be able to have that type of comparison. So I'll just leave you with a small vignette of some recent work that my group published, where we looked at, we were fortunate to receive a human embryo, which was roughly 17, 18 days after fertilization. So it's really very early, just 3 or 4 days after implantation in the womb. And we were able to characterize all of the cells, and then using computational approaches, the computational work in collaboration with colleagues in Germany in Munich, we were able to make inferences about how one cell becomes another cell. And that is a fundamental importance in developmental biology and also in the context of regenerative medicine, because of course, all the billions of cells in our body come from the division of a single fertilized egg. And that the cells initially are relatively similar, but then they start to diverge and become different from each other. So if we want to be able to build cells, tissues, entire organs in a dish, we need to be able to understand how cells change and how, because of course we could take the brute force approach and try random things in a dish in vitro to try to make one cell into another, but it's much easier to try to learn from nature. And you do this in engineering as well, in designing, suits you can swim in better, for example, or wings which are better for flying, more aerodynamic. In biology as well, of course, you get to learn from how it occurs in nature and then apply those in a regenerative clinical context to try to generate different cell types. So that was just to I guess, give a brief overview of why fundamental research is really important and why fundamental research will continue to play an important role in the IDRM within its vision and the goals of furthering regenerative medicine. So I'll hand back now to Paul and George to tell you a little bit more about this.
Thanks. So I think the key word in here was to understand how to regenerate. There is, from a medical point of view, and particularly as a pediatrician, there is one particular aspect that is also coming into play here, and that is diseases, developmental diseases, malformations, for example. In order to understand them completely, we need to know when do they occur, at what developmental stage, with which programs, and then that understanding will guide us basically with attempts to use regenerative medicine to overcome such a defect. So, for example, in the immune system, there's a particularly important gene, actually twins of genes called recombinant recombinase genes that allow the cell to form a receptor, which is essential for the cell to survive, be it a T cell or a B cell. And in the absence of this gene, the cell can't develop, and one has a block in development, and the children are born basically without an immune system. Now, in some of these cases, there is a bone marrow donor, and the individual can be cured by a genetically identical, with the exception of the defect, genetically identical bone marrow donor. That sometimes is very difficult to find a so-called match, an individual that is genetically so closely related to the patient that there is no rejection response and no consequent disease such as graft versus host disease. And one of the ways to overcome that is by providing the gene itself to the patient, the defective gene itself to the patient, to then allow that cell with now the corrected gene to develop normally. Only when you really understand when the gene is turned on, how it needs to be and where it needs to be turned off, you can provide the right correction. And so developmental biology has a lot to do with pediatrics and holds the key to understand disease, but also to very specifically provide the solutions for curative therapeutic efforts. So this is how I, as a pediatrician, I'm exactly seeing that IDRM can provide further knowledge at the level of how diseases come about and how we best can cure them.
Thanks, George. So just in the remaining time, I thought I'd just give you a little bit of potted history and an update on where we are with the new institute. So it's 2022, remarkably, and for George and I, that is a 10-year anniversary of a conversation that literally was held in the Jesus College Senior Common Room, where we discussed the possibility of a potential institute, initially focusing on developmental medicine, which was entirely appropriate for some of the skill sets and some of the people and research groups, George's interests, my interests, Shankar's interests, and others in developmental biology and pediatrics, as George has mentioned. And we sort of sat on that idea for a little while, and then we sort of thought about it a bit further, and we were summoned to the hierarchy at the Medical Sciences Division and the university to think about it some more, because there was possibilities for fundraising and making this a reality. And we thought about putting in the component of regenerative medicine because it was a natural lead on from developmental biology, as we've been describing throughout. And as soon as we did that, the powers that be that could get behind us and provide us some support to try and fundraise, and I should say that the project had £0 against its bottom line to begin with, and we were tasked to try and raise the money and move forwards for what is and is now materializing a purpose-built research institute on the Old Road campus in Headington in Oxford. And as we sit here today, some 10 years on, we successfully raised what is £35 million, which includes university contribution as well as high net worth individuals, etc. And the British Heart Foundation, a very important Japanese donor, Dr. Nakamura. And we have got to the point now where I'm pleased to say that a week tomorrow, we will be going around the institute with a view to hopefully signing off and the university accepting the building from the building contractors, and then with a progression towards moving in around May time, we would think, once all of the testing of systems, etc., has been put forward. So just to give you an idea of the building itself, it's 6,000 square meters, it's 3 floors. It will house 240 researchers. There will be around 15 to 20 research groups, depending on obviously size per group. The 3 key themes are neuroscience on the ground floor, cardiovascular science, in which myself and Shankar will participate on the first floor, which is mainly BHF funded and resourced, and then an immunology floor that George will lead on the second floor of the Institute. And within the Institute, we will have high advanced imaging capability within a designated imaging suite that Shankar has mentioned the importance of imaging in his work. It's very important across all biomedical sciences, and we will be building on that with a very nice collaboration with the Kennedy Institute for Rheumatology and Inflammation on the same campus site. And we've formed a collaboration with Zeiss, a world-leading manufacturer of advanced microscopes. So that's extremely exciting for us moving in. We will have omics technologies to be able to do the most cutting-edge, state-of-the-art sequencing of cells, both having isolated those cells, as Shankar alluded to, from the very early human embryo, but also in situ, so in tissues, using something called spatial transcriptomics that will be available to us. And we are also hoping to build links into mathematical modeling of regenerative medicine, developmental biology through the use of artificial intelligence, machine learning, and obviously mathematics. So reaching out to colleagues around the university to assist with that and having a hub of activity in the institute. And of course, this will link directly into Jesus College and the digital hub, Northgate House project. So we're very excited about that. And the plan, of course, is to try and bring in studentship programs in this area, which we would hope the students would then be based in the middle common room and beyond at Jesus College. So this is an exciting initiative for us, and we're very delighted that it's come virtually to fruition. Obviously the key aims, I think George, Shankar, and myself have already alluded to this. We want to find therapy. Let's back up a minute. We want to do cutting edge, world leading research, and some of that will be basic discovery research to make inroads into some of the problems and questions that we have around how do cells become specified, how do they differentiate, how do they form tissues, how does the immune system interact with tissues, how do they change during disease, during injury, what happens in development that we can mimic in adult disease, also birth defects, how do we correct those, what are the problems that occur there, fundamental questions, fundamental research, but obviously leading to a pipeline in which we develop drugs and new therapies to improve outcomes in patients, both in children with birth defects and in adults with acquired diseases of the central nervous system, the brain, the cardiovascular system, and the immune system as our primary focus. And just a point around the design of the building as an interesting concept from the architects is that they have given us vertical movement as an important part of collaboration and multidisciplinary approaches, so that we're not sort of working in little silos within these key areas of science, but we will be moving between the 3 floors, interacting with each other and working together to understand how the brain and the heart, for example, how they operate and how they might have similarities but clear differences. And we're particularly interested in intersecting with George's area of expertise, because understanding how the local injury and disease environment is affected by the immune system, and then the scarring that George alluded to and fibrosis afterwards is really important. If we don't get a handle on that, we're not going to be able to regenerate anything. And I think that's part of the reason why some cell therapy trials have largely failed, because they haven't dealt with the problem of the rather hostile environment, the inflammation and scarring that occurs. So we've got angles into both restoring tissue, cells, tissues, but also thinking about how do we condition this local environment. So that's a sort of, in a nutshell, where we are with the Institute. And as I say, it's extremely important that we retain strong links with the college, because the idea was formulated in the college. We are all 3 fellows of the college, and we are very keen to maintain strong links with all of the exciting developments, not just around the digital hub, but some of the aspects and the fundraise and the aspects that Brittany and her team lead in terms of focus calls for funding that we can tap into as well, to build a really, really cohesive relationship and collaborative and relationship, making use of students and so on that could work between the 2 sites. So very, very exciting. I think we're ticking down to 1 minute. So I don't know if there's any closing comments from George or Shankar before I think we're going to take questions.
Yeah, I might just add to not how we came about to meet in the senior common room on a cold grey day, but how we basically thought, coming from different corners here to creating the Institute, what is missing? For the Department of Pediatrics, it was missing this link to developmental biology, to understanding how pediatric diseases can have and frequently do have an origin in development gone wrong. But we also realized that actually this concept of marrying research that is built on developmental biology using as an output regenerative medicine, and using regenerative medicine underpinned by an understanding of developmental biology, that this was very unique. And when we looked across the globe, with a lot of fantasy, I think we identified 2 other places that were slightly different, but this combination will be a unique thing that I'm sure will be copied with over time as we go forward, and can use these 2 systems in an integrated way to create better therapies.
Thanks, George. Anything to add, Shankar?
No, I think that was a good wrap up. And so if, hopefully people have questions in which we can have a bit of a discussion.
Great, thanks.
Thank you so much, all of you. And it's very exciting also to see the opportunity maybe that the Jesus development might be able to help, provide with the digital hub and bring some more opportunities together there to share research and also to just showcase a bit more of what's going to be going on in this fantastic new space and the research you'll be doing together. And also just very exciting even to hear about the layout, allowing you to collaborate together, forcing those interactions to happen, whether you want to or not, of course you do. We do have a question in the chat from Brad. Brad, I don't think this is an elementary question, but Brad asks, how can your work on the epicardium help to restore heart function post an MI? Who wants to take that?
I guess I ought to.
Oh.
That's typical of Brad, right to the point and absolutely spot on. So Brad, the answer is in the current work we're doing around the human cell screening. So when we did this work, it was in a mouse model of myocardial infarction, heart attack, and we had a particular compound that was called thymosin beta-4 that stimulated epicardial cells to proliferate, so expand in number, migrate actually to the site of injury. We have no idea what the guidance signals are that made that happen. It was quite remarkable. And then they differentiated, as I say, into cells that contributed to new blood vessel growth, which of course helped the heart enormously, and also to fibroblasts, which were not scar-forming fibroblasts, but fibroblasts that help the skeleton of the heart being restored and the matrix of the heart restored around the area of damage. So a remarkable effect. And although thymosin beta-4 itself is not a good drug, it has a very, very, very short half-life. What we've been doing is then taking patient-derived epicardial cells from people undergoing coronary artery bypass surgery, making primary cell cultures in dishes, and have made that high throughput, so scaled that up to many, many different wells of cells. And we've been using chemical libraries of known drugs to see if we can affect the activation of those cells. And the activation is restoring developmental programming. So it's making those cells behave like they used to do in the embryo during pregnancy, making them divide, making them migrate and become activated. And there is a way of determining that through a process of which they undergo a transition. And that is our readout for a drug screen, a phenotypic drug screen using known drugs. And the reason we use known drugs is because, of course, they've all been tested for other indications, other diseases, and it's a more accelerated way of potentially repurposing those drugs for potential treatment. So that's where we're heading with that, and it's very exciting. And we've been moving that forwards with some chemical matter that actually does activate these cells in these known drug libraries. So we now need to do the validation and push those studies forwards. But great question, thank you.
Thank you so much. Chris asks actually a question that's hits quite close to home for me. So how do you see the future of dementia, Parkinson's, Alzheimer's, etc. in terms of regenerative medicine? Is there hope? Could regenerative medicine make a positive impact? My father had Parkinson's, so I would quite like to know, is there hope for others?
The answer is basically on the principles of what we try to achieve, and I can't give you, I think, sufficiently knowledgeable answer how this relates then to particular neurodegenerative disorders. But by knowing how do stem cells in the brain behave, and they do exist, and they are there, and what are the molecular cues that they are required to develop in certain lineages to attain certain functions, will clearly open up an opportunity to recapitulate development even later in life and repair cells that have acquired a program that makes them ill and that renders them with limited or even actually a loss of function. So while the brain in particular is a very difficult organ for regenerative medicine, particularly in the way how it has been done in the past, with either injecting cells or trying to manipulate resident stem cells, we will in the future see with a better knowledge where these stem cells sit and how their dormancy can be changed to active development and recapitulation, what they had done during embryogenesis, for example, that with this knowledge, new therapeutic opportunities will emerge. Now, how quickly will that be? That is another question from Paul, I think, yes. Very difficult to say. I think, you know, I would think also for IDRM, for things which are a little bit more tangible, we're thinking of 8 to 10 years until some of the principles that we work on will be translated into a medical opportunity that can be tested in a clinical setting.
Thank you so much, George.
I don't know whether Shankar or Paul, you have anything more specific to say in this context.
Well, I would only add that there is activity. Of course, we're not the only ones trying to tackle these very important diseases, and there's a very, very prominent group within mine and Shankar's host department, Richard Wade Martins, who's looking at Parkinson's and is heavily funded by the Parkinson's Disease Society. There are groups actively studying this that will not be based in the Institute, but of course, people we are in close contact with and could potentially collaborate with. And Richard Wade Martin's group are looking at ways in which human pluripotent stem cells can give rise to neurons, etc., that could be used for therapeutically in diseases like Parkinson's, and have been working in that area therapeutically for quite a while now. So there is promise from their research program, and they've just moved into the new Kavli Institute, which is literally opposite our department as we speak, which is a very exciting new development in cell biology and various disease processes. So just to add to that, really.
Next question. Okay, so James said that mathematical modeling was briefly mentioned. Do you anticipate a role for mathematical and computational modeling in your work?
Yeah, I can answer that briefly, I guess, from the outset. So George and myself in particular were very keen to get this element into the Institute, because we can learn an awful lot from modeling cellular and disease-related processes. And we reach out to colleagues such as Sarah Waters and Ruth Baker in the Maths Institute to help with this aspect of the work. And just to give you an example of an ongoing project that we've been exploring, we're modeling scar formation or fibrosis in simple terms. So we take activated fibroblast cells that can lay down matrix that is scar, collagen that is scar, and we can model how that looks through changing behavior of those fibroblasts. So what happens to the scar if you make those cells migrate faster, or if you make them divide when they're actually meant to be non-dividing and just laying down collagen? What happens if you challenge them with an inflammatory cocktail of molecules that might put them into a more injury, disease sort of scenario? Is good for us, because what we can then do is test a a drug that might influence the scar. So as George said, one of the things that is important to understand is how you can resolve the scar over time so that you can then regenerate the tissue. So if we can predict the outcome of an intervention of a drug by modeling that process based on our observations on the cell behavior of the cells that actually lay the scar down, then that gives us real hope of moving quickly into testing those drugs then in the necessary in vivo animal models. But the modeling element is extremely important. And using things like machine learning to get more parameters where you understand more of the variables of the cell behavior than we ordinarily can determine by the naked eye or by a human operator, that sort of thing is a really powerful approach. So we are very keen to establish that and have some ongoing projects in that area. Shankar, do you want to add anything?
Yeah, absolutely. Thank you, Paul. In fact, I can give another, in a sense, a very different example. We've used mathematical modeling to look at in the context of morphogenesis, how the shape of organs and tissues emerge. This has been work in collaboration with Ruth Baker, who Paul mentioned. And there you represent individual cells as geometric structures, and you computationally try to determine the forces that are applied to each of the cells during cell movements. And this can really help you try to understand how interlinked cells in a sheet of in a tissue, like or what is called an epithelial sheet, which is comprised of cells that are all linked together, how you can get the movement of individual cells within such a sheet, or how you can get the deformation of such a sheet into the shape of a structure. So absolutely, mathematical modeling I think plays a fundamental role in all of our research. And computational approaches as well. As Paul mentioned, machine learning approaches are so almost not common, but they're pretty widespread. And we've used those type of approaches to try to get the outlines of cells automatically so that you can then try to compute the volume, physical parameters such as the volume, the surface area, the curvature of cells and things, and make inferences about the forces that those cells are subject to. And we can, of course, also make actual measurements, real measurements of the actual forces, and that can help you build better models and also test the validity of the models. So absolutely, I think there's a lot of room for interface between biology and mathematics here. So one small point to also add from the college perspective, that's one of the beautiful things about college, where we can get have discussions with people you may not expect to have discussions with. And so I've had discussions with Jim Oliver, who's the maths fellow in college, and over lunch. So it's the typical story everybody talks about, or sitting down for lunch and just by chance talking about our research, and then it turning out that Jim had very important insights from his work on fluid mechanics to offer to us when we think about how cells move in the context of development.
Great, and I think there was a question that might have tied into an earlier question, so I don't know if Paul Bartlett thinks that this has been resolved with some of the earlier conversation, but he was specifically mentioning progressive MS and the institute may be having an effect on that treatment. And I think, George, you were sort of saying that the research will come into action or into medical practice probably 8-10 years down the line, but did you want to say anything specifically related to MS, if you could?
I can't, to be honest, without saying, very general. We will have on the ground floor of the institute people who are interested in the neuronal system, and also will look at rare neuronal diseases, mostly in a developmental context. But how well these kind of diseases become then models or examples where you can gain a lot of understanding that's relevant for other diseases that typically occur later in life, such as MS, is not known yet. From an immunological point of view, MS is obviously particularly important and interesting to know because it is an immune response coupled to MS that destroys the normal structure and composition of the central nervous system, and understanding how the immune response can be deviated from a destructive to a possibly bystander presence or into a condition that allows regeneration to occur and favors regeneration. That could be one therapeutic approach that I know people are working on it. But again, here at the bottom of the overall challenge is what triggers the disease and how does the immune system lose its competence to distinguish between self and non-self. And that almost leads a little bit over to Paul Williams' question with his question concerns, so if you allow an immune system after birth to develop normally, and the immune system now sees a lot of tissues, all of our tissues in the body, would there not be a danger to develop autoimmunity? And this is a very central question, and a lot of people have worked on answers to this. And just to make it very short, the immune system is taught to distinguish between beneficial self and injurious non-self, in part in the thymus, which is this organ sitting on top of the heart. And in the thymus, there is a particular type of cell that can express as a collective almost all protein coding genes that we have in our body. So what these cells collectively do, they have a library of peptides that represent almost all of our body's own proteins. And little snippets of these proteins are being used to educate the immune system. And normally this is very comprehensive. It occurs during development, but because the thymus is there and develops after bone marrow transplantation or after the repair of an immune deficiency due to the example that Paul gave to a repair of RAG1, RAG2 genes, this capacity to educate immature T cells to know exactly that I can't attack because this is my own body's proteins, that is maintained. And with a few rare exceptions of diseases, congenital diseases, is actually there throughout life. And so reconstituting the adaptive immune system does not necessarily mean that you end up with severe autoimmunity.
Oh, that was great. And I thank you for looking after Paul Williams's question, because as a historian, it was really testing my limits. So maybe just to go back one question before, what hope do you have that your work will extend in the future? What wider collaborations do you anticipate within and outside the university? Is it likely, will be likely to be in place in the future? What do you think?
Well, I mean, I'll come in on that one, and we must answer Ingrid's question as well, because Ingrid's question actually gets to the premise of why I use developmental biology. So we could close on that. It would be a perfect point to close. But just to comment on the first one, which is a great question. I mean, we've already mentioned that, you know, we are looking to produce new therapies. We are studying fundamental discovery science at one end of the spectrum, and that will lead to opportunities to translate those findings into more clinically relevant drug-based or tissue-based repair strategies. But I think the wider collaborations come from utilizing the local ecosystem at Oxford, which of course is second to none, around multidisciplinary approaches. So we've already mentioned colleagues in the Maths Institute that we either work with or have worked with that we'd like to bring work more closely together around modeling, computational biologists around, and the use of AI and machine learning. That's a very big area that we'd like to get into and expand into. We think that's very important for making lots of predictions about the biology we're looking at. And I think engineering is an important component. So bioengineering, and of course we have the Institute of Biomedical Engineering on campus, on the same campus in which we hope to forge strong links with. And then with respect to taking forward further interactions with more immunologists, because immunology is really important, the Kennedy Institute of Rheumatology Inflammation. So actually making use of the local ecosystem. And then as we generate large data sets, which we will do through our imaging experiments and our sequencing experiments, then we have the perfect institute on our doorstep in the Big Data Institute, which is able to handle, analyze, and interpret data sets like this in a way that's really second to none. So I think broader collaborations will drive forward the more sort of translational work. And just to come to Ingrid's question, so that question, if I'll just read it out, is given that you're angling your research towards translation, which we are, as we've said, albeit it might be some way down the line, is it a concern that regenerative medicine could go too far, lead to hypertrophy, cancer, etc.? And how much research will there be at the new center addressing this? Well, this is why studying developmental biology and understanding how development makes organs in the first place in a very, very regulated manner in which you do not get cancer, you do not get, you get a transient hypertrophy in some organ systems, but it's regulated and brings the organ back to fully functional, is precisely why, or one of the key reasons why we're doing this in a regenerative medicine capacity. Because if you think about it, as Shankar's alluded to, the embryo forms with an incredible pattern and regulation of cell behavior, tissue formation, etc. It's all under control. That, of course, it does go wrong in some cases, and there are instances of childhood cancer and there are instances of birth defects, as we've alluded to. But the normal developmental processes are precisely what we want to understand so that we avoid the issues of cancer, etc., in our targeted approach in the disease and injury setting. So I think this is a great question because it really highlights why studying developmental biology is so important in this context.
This was a very good summary why understanding how the normal system evolves and controls itself for normal shape, normal function, is the key to prevent that a aberration, a derailment of development occurs and leads to disease. And frequently I try to explain it in the way that we probably, or many of us, have experienced when we were children. We found a watch and we took it apart. Just want to see how it works inside. And then we put it together again. And miraculously, 2, 3, 4, 5 pieces did not fit in the watch. And we just put it aside and we closed the watch and it didn't work. For various obvious reasons. And so only once we know where all the elements, how they interact and how they play, we can really repair things properly. Otherwise, our approach is, I think, just for the sake of the complexity that we try to understand, is just not good enough. And defective regeneration will occur. And that's clearly something we want to avoid with the knowledge we generate at IDRM.
Thank you so much, everyone. And thank you so much, Paul, Shankar, and George, for tonight's session.
Thank you.
Thank you very much.
Bye, everyone.
Thank you. Bye-bye, everybody. Thank you.